ahead and introduce our program. And thank you all so much for attending. So welcome to Popping the Science Bubble. This is a monthly seminar series that aims to share new research findings from grad students and postdocs at UC Berkeley with the general public and create constructive discussion about a variety of science topics. We have 2 speakers at each seminar who will talk about their current research or a topic that they find really interesting. We have had so many interesting topics over the years and the months, and I encourage you to keep checking in and seeing what we're talking about. We update our calendar, our online calendar, and also Popping the Science Bubble is sometimes advertised in our Berkeley Public Library email listserv. The organizers of this program are 3 graduate students at UC Berkeley, Jenna, Madison, and Oksana. If you're interested in checking out their past seminars, you can visit their website, you can check out their Twitter or Facebook, and sign up for the listserv to keep in the loop for upcoming seminars. There's also a great Popping the Science Bubble YouTube account, which since COVID, we've been doing this program digitally, so you can see the past videos. I'm going to hand it over to the Popping the Science Bubble team to introduce today's speakers.
Hi, thank you, Kelsey. So we're really excited for the talks tonight. As Kelsey said, we're a monthly seminar series. We host graduate students and postdocs who are researching in the Berkeley campus and affiliated research institutes. We give a talk every 3rd Tuesday of the month at 5:30, and we advertise on Facebook and Twitter and over email if you're interested in hearing more. We also have a website. And so our first talk of the day today will be, I should mention these talks are open format where we're taking questions from you guys. Feel free to please enter questions as the talks go in the Q&A and the chat, and also in the comment section on Facebook. We will be monitoring all of these so that we can help you guys engage with our speakers virtually. So with that, I would like to introduce our first speaker, Evan Groover. He's a PhD student in the Plant and Microbial department here at UC Berkeley, and he's studying how plants and bacteria take carbon dioxide out of the atmosphere to create biomass, and how understanding how this process works can be leveraged to make more productive and sustainable crops. Along the way, he's also developing tools to modify crops for increased yield, disease resistance, and nutrition. And outside of his research, he is a jazz saxophonist and regularly performs around the Bay Area with a few different musical groups. So we're happy to have you join us, Evan, and please take it away.
Awesome. Thank you so much, Madison. I am so excited to speak with you all today. Thank you for coming out for a talk that is titled Sequester This, a critical look at humanity's hopes of removing CO2 from Earth's atmosphere. So again, my name is Evan Groover. I am a plant biologist and a gene editor, and I'm currently a PhD candidate in the UC Berkeley Department of Plant and Microbial Biology. And so just to give you some context on why I'm here today doing what I'm doing, I study photosynthesis. So photosynthesis is the process by which plants and algae and bacteria take carbon dioxide out of the atmosphere. And so what specifically my research focuses in is sort of manipulating the molecular mechanics of photosynthesis in plants such that we can kind of improve this process of carbon dioxide assimilation. And that topic specifically is not what I'm going to totally fixate on today. I'll explain what I'm doing, but it came up in this talk, in preparing for this, that we're in something of a special place in Berkeley, And I just wanted to point out that when we talk about CO2 and the, the sort of the mechanics of CO2 assimilation, it's always sort of prescient to point out that, or, I guess notable to point out that here in Berkeley, we're kind of sitting at the helm of photosynthesis in some capacity, that the Calvin cycle, this chemical or biochemical process by which plants and algae take carbon dioxide out of the atmosphere, was actually first characterized here at Berkeley. And ever since then, some of the most foundational advancements in photosynthesis research and also CO2 assimilation research have occurred here on campus. And so it was Melvin Calvin, his postdoc Andrew Benson, and James Bassam back in 1950 that actually first discovered the mechanics by which plants were able to assimilate carbon dioxide. And sort of unfortunately, in my opinion, it was Calvin that solely won the Nobel Prize in chemistry for it in 1961. But all three of these men have had, a sort of indelible impact on plant biology and sort of our understanding of sort of the Earth's biogeochemistry at large. And just a fun fact, because I know that I'm talking to Berkeleyans, if you are in the southeast corner of campus and you see a big round building that looks like this, that's actually sort of the vestige of Melvin Calvin's laboratory. It's now, an institute for computing, but they've seen that it's been preserved, as best as it can be to still kind of bear witness or bear testament, I guess, to the really seminal biochemical work that took place there. And so I study photosynthesis and I study carbon dioxide. And so you might imagine that I spend a lot of time thinking about this graph. And this is sort of our, kind of recent geochemical history of the Earth with regards to atmospheric CO2. So if you look back over the past 800,000 years, you'll see that CO2 levels have stayed within a pretty predictable range, with some kind of periodicity that can be expected with cycles of the Earth. But in the past 150 years, there's been this really precipitous spike of atmospheric CO2 that has been deposited as the result of burning fossil fuels. So this is almost entirely owing to human activity and industrialization. Now, taken at face value, I always find this kind of abstruse, so I think it's kind of important to consider the broader carbon cycle and really what is CO2 and where does it come from. And so I always like to show this image that shows sort of the carbon cycle at large. So each one of these black dots is a gigaton, a billion tons of carbon that are just kind of circulating around. And essentially, in a steady state equilibrium, carbon in our atmosphere and in our environment, in our biosphere, is kind of moving between the ocean, the deep ocean, the biosphere, living organisms, and the atmosphere. But as we start to move towards the recent decades, what we see is that humans start really precipitously burning fossil fuels. And these fossil fuels are emitted into the atmosphere, and what has actually happened is that they've overwhelmed the biosphere and the ocean's ability to act as a carbon sink. And as a result of this activity, carbon has started to dwell in the atmosphere and form these sort of greenhouse gases that we now know have sort of a warming effect, a heating effect on our Earth's atmosphere. And so this is the situation that we found ourselves in. We've burned a lot of fossil fuels, and we're, in a position that it's starting to have some pretty nasty effects. Sea level rise, major deforestation and habitat loss, all sorts of ecological upturns, and then really extreme surface temperatures, both hot and cold all over the Earth. And so it stands to reason that if these emissions are to continue to go unchecked, then in the coming decades, we're going to see really, really insane things with regards to human and environmental impact. And so in response to this threat, and I guess an disciplinary and intergovernmental panel of scientists kind of pooled their expertise, and they came together a few years ago to say that, there should be kind of a set number of degrees in temperature that would be permissible for human society. And they decided that, right now we've probably heated the Earth about 1 degree Celsius on whole, which is a ton if you actually consider, the entire Earth and how that's sort of a mean value. But also that if we're to exceed 1.5 degrees Celsius, it's going to get really bad. And that we as a species and as a society need to focus our efforts on keeping, this greenhouse gas borne heating below 1.5 degrees Celsius. And to do this, we don't just have to cut our emissions. We do have to cut our emissions really significantly, but we also need to be thinking proactively about how we can sort of take out some of that CO2 from the atmosphere. And so in recent years, there's been a really strong interest in developing what is called carbon direct removal technologies, or CDR technologies. And these are technologies that essentially sequester carbon dioxide from the atmosphere, from open air, and then are able to store them in some very stable, usually underground space or form, such that they're not able to be re-emitted, excuse me, back into the atmosphere. And so I admittedly do study carbon interactions, but I probably, like you, hadn't spent a lot of time learning about all of these various technologies that could be used for open air capture of carbon dioxide and sequestration of carbon dioxide. And, you know, I knew about them and I learned a little bit, but I didn't really have a strong interest in learning about these things until about January of this year, when the Biden administration announced that they were all of a sudden very interested in carbon removal tech. And, you know, me, the savvy plant biologist, thought, well, what's a better carbon removal tech than a plant, right? You know, surely they're just going to give us all the money that they're talking about. But that actually wasn't the case. I saw the Australian government making similar pledges with money that they wanted to start investing in carbon capture technology. Then I saw major corporations, Microsoft, Amazon, and Google, pledging, you know, on the order of billions of dollars towards, carbon removal technology. And then, entrepreneurial / trolls going on the internet saying that they have, a bunch of money that they'd like to invest in carbon sequestration technology. And so I thought, man, what the heck? What are these people actually going to invest in? What's the best game in town if you want to pull out carbon dioxide from the atmosphere? And so I've been doing a few months of research and following this space, and trying to apply what limited expertise I have so far to considering this question. And mostly what I've done in preparation for this talk is I really have kind of been scrutinizing the portfolios of these governments and corporations and large non-governmental organizations that have been investing in carbon capture, and trying to parse out how feasible these technologies are and what they could do with regards to kind of remediating our atmosphere. And so today I'm just going to give you a sort of a talk about that. So I kind of want to just outline, to my eye, what are some of the most prominent technologies that are currently being implemented for carbon direct removal. What are the relative strengths and weaknesses and sort of feasibilities of these technologies, and how much they actually would cost to scale to something that could have a planetary or atmospheric effect. And then I kind of really want to talk about, where each of these are with regards to, their technological potential, because some are certainly more advanced or more feasible than others. And then, since I am a plant biologist, and, I would love to talk about anything that I have a little bit more training and expertise in, I'm going to spend a little bit of time at the end just talking about how plants, and specifically how, engineering in plants could play a role in direct carbon removal from the atmosphere. And so, to my mind, the technology that makes the most sense for removing carbon from the atmosphere is just forests, right? Trees are, really good sequesters of CO2. They turn CO2 into forest, which is, presumably a really good carbon sink. And I think a lot of people think the same thing. And in fact, some 30% of the emissions that humans put out are actually sequestered in forest already. So they already act as a sort of effective sponge of human industrial emissions. But it's no surprise to any of us that, this is sort of a diminishing effect as humans are expanding, converting more places into arable land for agriculture and livestock. Our forests are getting cut down pretty significantly. And so this is sort of a historical representation of what the Amazon looked like, and then a modern day representation of how much actual forest is acting as a lung for our planet. And so this is a pretty grave threat to the sink that we have in the biosphere for carbon. And so over the past about 21 years now, ever since 2000, all sorts of NGOs and governments have been pretty significantly investing in afforestation technology. And so this is and I should say maybe afforestation and reforestation. So afforestation is the process by which you take a land that hasn't traditionally been a forest, usually a place that is like for waste or sort of is barren, and then trying to establish a forest there. And obviously the approach here is pretty varied based off of the climate or, the ecotype that you're working in. But afforestation is, up until this point, pretty much the most significant climate remediation technology that has been put in place at scale. We can see here, this is just from 2015, but it's some of the best data that compares, the variable governmental kind of investment in this technology. We can see that China, who, almost sort of surprisingly is sort of much more quick to acknowledge the severity and the importance of meeting climate challenges, has invested very significantly and is continuing to in afforestation. And so this is sort of something that, it's sort of hard to track. I've learned as I've really tried to drill into the data of this, it's like there are a lot of people that are invested in trying to plant forests all over the place. But it's very evident that the governments of the world have been investing pretty significantly over the past 20 years. Something that I was kind of surprised to learn about in this is something that I had heard of but not really understood, and it's called the Albedo Effect. And it's actually this concept that though it's nice to plant forests and they act as a carbon sink to actually sequester atmospheric CO2, in many cases, there are some cases where you don't want to plant forests because forests are actually too dark in color, especially in glacial areas, in boreal climates. And which is actually like kind of the predominant ecotype on our Earth, if you think about it, is boreal forests. Snow acts as like a significant reflective force as to stop the absorbance of the sun's rays. And so when you actually plant more trees in these areas where there would traditionally be tundra or snow, you are then kind of increasing the amount of solar radiation that is absorbed in those areas. And as a result of that, you have a greater kind of effect on just warmth absorbance, and this is a totally a non-desirable effect if your goal is to mitigate global warming. And so actually afforestation isn't really a cure-all. It actually only works in certain environments, and the field, I can assure you, is rife with controversy about where we should be doing afforestation and reforestation and where we shouldn't, and experts don't necessarily agree, but when do they ever? So far.
Hey, Evan, sorry, quick question about your graph 2 slides before about trees.
Sure.
Yeah. So do you know how the data is collected? Is it self-reported, or are there like satellite images?
So from this graph, I know that it was self-reported, but yeah, I think it's sort of a really hard thing to keep track of. So whoever is asking this question, I think maybe if you're at all skeptical, I am too. So maybe but I think there are some very public and sort of demonstrative examples of afforestation that have occurred in China, and so that's it stands to reason that this is legit. So what's cool about afforestation, it really involves no technological advancements. Humans know what forests look like, and we are pretty good at making new ones. And also forests are like absolutely critical for, especially reforestation is going to be critical for ecological and environmental conservation, which if you're like me, that's a huge priority. But this is getting tougher as we kind of advance as a species. In 30 years, we're going to have 9 billion humans, and those humans need to farm and they want to eat cows, and so they want to have clean water, and they're going to be competing for resources with places that we might put forests. Also, forests are very vulnerable to pests, to fires, and to drought, and so it's actually there are many situations where, because they are so vulnerable, you might imagine that they're actually going to be carbon sources if they're not properly maintained. And so that's a huge risk. And also things like the albedo effect, biophysical feedbacks, just simply because they're not so understood, they do pose something of a threat. Afforestation is the cheapest technology of almost all of the carbon sequestration technologies that we know of. And so that's kind of a nice take. And so far, almost because of that, forestation strategies really make up the bulk of current carbon offset strategies. The US invests really significantly into forestation techniques, and especially the state of California, who issues these carbon credits. Most of them are going to people that are doing reforestation and afforestation approaches. So probably the second and the most hi-fi, like science fiction-y type thing that people are considering in the carbon sequestration space is direct air capture. The direct air capture approach is sort of a chemical engineering approach that was born of coal burning coal burning plants, usually these places that historically produce all sorts of CO2, and this push for clean coal. And so this was from 2014, we really developed the first truly clean coal plant. This was up in Saskatchewan. And essentially, this is a technology by which you use some chemical approaches at capturing CO2 as it leaves a plant, and then sequestering it, and then storing it deeply in subterranean, usually geological spaces. It was just in the past few years that people started realizing, well, what if we took those tools that people were using on coal plants and we just put them in the open air and use them to capture CO2? And this is a pretty significant technological challenge. I mean, the air that's leaving a coal plant is sometimes 15 to 30% carbon dioxide, and our air is what, 400 ppm, so it's like 0.04% carbon dioxide. And so it's sort of a huge technological challenge to actually capture CO2 in open air using chemical engineering approaches. But people are still trying nonetheless. This is one of the first direct air capture plants that's out in Switzerland. It's a company called, can't remember what it is right now. You'll see it in just a sec. Essentially how this works is they have these huge fans that blow air super fast, and they have these materials that capture CO2, and then they have to heat those materials to like 1600 degrees Fahrenheit to release that CO2, and then effectively capture it and pump it underground so that it can be stored, and then they release the rest of the air that has been filtered out just back into the atmosphere. So sort of a really large scale chemical separation, if you think about it. These plants are absolutely insane in the infrastructure required because they essentially require an accessory geothermal power plant. And so the platonic ideal of this is represented here. You can see that you essentially are you're capturing CO2 and you're pumping it underground, deep underground into these geological formations. And then the energy that is generated from that pumping is then captured by a geothermal power plant, creating this quote unquote closed loop of energy resources and carbon sequestration. This company from Switzerland, lest I forget to mention, is called Climeworks. If you're interested, this is a depiction of the plant that they've made in what's it called, Hinwil, Switzerland. So direct air capture is pretty high concept at present. In premise, it can occur anywhere, but it requires so many resources that's hardly the case right now. Proponents are really quick to point out that it might create hundreds of thousands of industrial jobs to be creating these plants for CO2 capture all over the place. But it's extremely expensive. The technology is very young, and it requires an incredible amount of resources to set up plants like these, not the least of which are a renewable energy supply, which is, kind of hard to come by in a lot of places, geological structures for sequestration, and, a water source that can, maintain a plant like this, which is very water intensive. Another thing that's kind of interesting that I've come to realize is that, it would be really cool if we could be turning the CO2 that we're capturing into something of use, like a fuel or, some industrial chemical, but that technology hasn't really come to fruition yet. There are scientists at Berkeley that are working really hard to see that, this CO2 that we're capturing might be turned into a useful fuel or something, but at present that doesn't really make sense at any industrial scale. By far, this is the most expensive technology compared to, maybe the $1 or $20 per ton that you would get from a forest for carbon. This is like $200 to $600 per ton of CO2 sequestered. So right now, this is a super economically kind of unfeasible thing, in my opinion. But it stands to reason that at an economy of scale, that might be different. And so what's interesting, and the reason that I'm talking about it right now, is that direct air capture has received a ton of attention, especially from companies like Microsoft that want to invest in, large technological solutions to climate change. And also, our Congress is now actually considering multiple bills to start doing direct air capture. Okay, I see. Am I going until the 5 or the 10, or does anybody know how much time I have here? I'm sorry, I kind of lost track at the beginning there.
I think you've got about 10 more minutes.
Okay, that sounds perfect. Okay, and so with that in mind, probably the last exciting technology that I've been looking into, and actually one of the most significant technologies that has received public and private investment over the past few years has been soil carbon sequestration. So you might be surprised to learn that the soil of the Earth contains more carbon than the entire biosphere and the atmosphere combined. The soil is an absolute hotbed for carbon, but this has started to change as agriculture has really scaled up over the past century. I love showing people this image. This is some dude out in the American South, and he's like standing in a prairie field, and you can see this sort of native prairie grass and its super, super deep roots that are extended into the soil. And then immediately adjacent to it are these wheat plants. And you can see that they have just these super short and kind of crappy roots that don't extend very deep. And the consequence of this, as we've uprooted native plant systems and replaced them with agricultural systems, is that we're storing much less carbon in the soil. This has really come at the cost of a lot of carbon that would be naturally stored and kind of sunk in our soil. And so the data on this is pretty diffuse. Not a lot of people, I've been surprised to learn, do super long longitudinal soil studies. But from what I can tell from across the world, there's pretty good evidence that almost everyone is having trouble with a loss of organic carbon in soil. And so this is just one representative study from some high plains out in India where they grow rice. You can see that over the past 40 years, as their society has kind of adapted to industrial agriculture, they've lost a significant amount of soil carbon. And this effect is actually really incredibly pronounced here in the United States. This is one study that came out just this year, and they found that a third of the topsoil has been lost in Iowa as a result of industrial agricultural practices. And that's like our greatest asset for agriculture in the United States. So this is super shocking to consider, this loss of carbon. And this has really come to the detriment of agricultural yields. Carbon loss and degradation has really, really immense effects on plant yields. When there's less carbon in the soil, that means that there are fewer microbes that are able to live there. That means that there's fewer accessible nutrients for, in certain situations for plant roots. And so, this is, despite, the advances that we've seen from industrial agriculture, there's been a precipitous limitation in what we could be getting because we haven't been tending to our soil very well with regards to organic carbon. And so there's actually some really good ways that we could be kind of improving upon this. We could be practicing no-till agriculture where we're not constantly upturning the soil and releasing carbon back into the atmosphere. We could be using more cover crops that more effectively utilize soil carbon. We could be using way less fertilizer and heavy input agriculture, as is the norm in the United States, is actually probably the strongest causative agent for organic carbon loss in soil. And then we also might start doing longer crop rotations and just considering alternative sort of agronomic solutions to fixing our carbon situation with regards to ag. And so soil carbon sequestration is a great and scalable idea in many regards. Carbon stays in soil for a really long time. And like I mentioned, having better practices with regards to soil carbon might have major agronomic benefits, higher yield and less fertilizer use. But at present, agricultural change is really, really hard. It's very hard to convince farmers just about anywhere in the world right now to change any of their practices, especially, on account for climate change. And so at present, the implementation of this technology is kind of stalled. Also, if you follow the trends of land ownership in the United States, there's a lot of upturn right now with traditional, familial ownership of agricultural lands, and not a lot of understanding of how we might best implement these technologies for soil carbon sequestration. So there isn't really, those two things taken together make it really hard to have these continued generational investments in, keeping good carbon in our soil for farmers. On the whole, though, this technology is kind of promising, or these approaches are kind of promising because it's pretty inexpensive. But at present, there are some economic, cultural, and technological barriers really adopting all of these best practices for soil carbon sequestration. That said, though, the Biden administration has expressed a lot of interest, and Biden's USDA and Secretary of Agriculture have expressed a lot of interest in maybe issuing a subsidy or a carbon credit to farmers that are, effectively farming carbon. And so maybe this will change. And I, as an agriculturalist myself, remain really optimistic about that. So those three things, probably the biggest frontiers of carbon sequestration, and it kind of begs the question, what's going to happen? And I hope, as a person who would like to see us remove a lot of atmospheric carbon, that we do all of them and at scale, and that private and public entities invest significantly into these technologies, and as many of them as possible. But realistically, a lot of these technologies are super young, and it's going to take pretty significant, not coercion, but convincing to get people involved and kind of in wanting to build out these things, because not all of these things are cheap, and not all of them make strict economic sense. Besides actually in the amelioration of climate change. And so finally, I'll take the last maybe 3 minutes here just to talk about my stuff. I love plants, and I am a plant engineer, and I am really interested in how we might be using plants to kind of more effectively sequester carbon dioxide from the atmosphere. And so a story that I like to tell, and is sort of the stuff and substance of my research, is that plants perform photosynthesis, this process of pulling carbon dioxide out of the atmosphere, but they haven't been doing it for very long. And in fact, plants have only existed for about 500 million years. Algae and bacteria, on the other hand, have existed and have been doing photosynthesis for some 3 billion years by certain estimates. Maybe, I think I can already hear my lab mates maybe chiding me for that one. Maybe we'll call it like 2 or 3 billion years. But and as a result of them having been around and evolving and doing carbon sequestration for much longer, bacteria and algae have become really good at photosynthesis, and especially carbon acquisition. They've evolved these systems called carbon concentrating mechanisms, by which they're effectively able to pump carbon dioxide into their cells very effectively, and are able to have a flux of carbon assimilation that's much faster than what is typically achieved in plants. And so the savvy plant engineers of this day, not to be too self-congratulatory, but are interested in how we might take mechanisms from these bacteria and these algae and put them in plants to essentially speed up this rate of carbon assimilation in plant leaves. So one thing we really would like to do, just for holistic agricultural increase in carbon sequestration, is to take these carbon concentrating mechanisms and put them in plant leaves. We also want to, make plants have perennial life cycles, or have, kind of different agricultural lifespans, such that we don't have to till soil so much and release all that carbon like I talked about. And we would also like to make plants just higher yielding and have less non-harvestable biomass, so that we would limit agricultural waste and be able to grow more food on less land, so we wouldn't have to keep cutting down our rainforests. It's kind of similarly, got cut off here, crazy. We also would like to engineer roots that can resist decomposition, so we can store more carbon in the soil for longer. We want roots that can kind of maybe fix their own nitrogen, or have better efficiency with the use of nitrogen, so that we don't have to put so much fertilizer that degrades our soil. And we want plants that have deeper roots, like that prairie grass, so that they can kind of stick around longer and store all that nice carbon in the soil. And so sort of the conclusion that I've come to, and by no means, is mine the final take on this, but I think carbon direct removal is a really important goal for, our planetary community, and it will be in the coming decades. And while there are a lot of technologies, I think it's not, none of them is sort of the silver bullet. We need to invest in all of these technologies and see, which are going to actually scale appropriately and equitably across our species. And realistically, there's not really a way to think about the development of these technologies without considering significant public and private investment in them. And so it really behooves all of us to, put our elected officials in a position to be seriously considering these technologies in the future, is my public opinion. And yeah, I have to say that I've left this whole period of consideration a little bit more optimistic than I was before. I think that there's some really great work being done. We have probably, one of the most significant and dire challenge that our species has ever faced in front of us. But there's some exciting work to be done, and I'm stoked to see where it goes. So with that in mind, these are my references if you want to pause the YouTube video later. I would really encourage you to check out these latter two links. These are just for you, the carbon curious person, as they have really good information about these technologies at large, including those that I didn't mention. And if you want to talk to me later or ask another question, you can hit me up right there. Thank you so much.
Great. Thank you so much, Evan. We have time for, I think, 1 or 2 questions before Claire's talk, if anyone has any through the chat or Q&A. Otherwise, I think Evan will be open to answering questions throughout the chat or directly in the chat box. But if anyone has any questions currently at the end of the talk, we're happy to take them now. All right, maybe we'll have you answer through the chat then. Oh, sorry, one just came through the, Laura says, thank you for the great presentation.
Thank you.
Not a question, but a thumbs up. Okay, then maybe we'll move into Claire's talk. And Jenna, you can give the intro.
Hey, yeah, so Claire Magnani is a chemistry graduate student researcher in the lab of Professor Thomas Maimoni at UC Berkeley. Her research interests include the total synthesis of natural products and the development of novel chemical methodology. She received her bachelor's in chemistry and physics in 2015 at Harvard University in the lab of Professor Andrew Myers. And before beginning her graduate studies at Berkeley, she spent 9 months at the University of Cambridge in the UK, where she conducted research under Professor Jason Chin at the MRC Laboratory for Molecular Biology. She'll be joining the lab of Professor Stuart Schreiber in fall 2021 for postdoctoral research at the Broad Institute. Congratulations. Claire was born outside of Paris, France, but grew up in Andover, Massachusetts as the eldest of 5 siblings. She repeated kindergarten since she did not understand a word of English during her first go. Outside of research, Claire spends time with her husband, Chris, and 7-month-old son, Julian. They especially like to go on hikes in the Berkeley Hills. And with that, let's welcome Claire.
Thank you. Maybe I should have started sharing my screen ahead of time. Sorry about that. Thank you for the kind introduction. And thank you to the organizers and Evan. Wow, what a great talk. I learned a lot. And I can't wait to delve more into that. I feel like my talk will be a little bit more niche, but I hope it'll open up some of our viewers' eyes to, what chemicals, the different ways we can consider chemicals, because sometimes chemicals just sound like this big scary thing you want to avoid. So my talk is entitled Nucleotide Mimics, Can Synthetic Chemists Imitate Nature to Make New Therapeutic Medicines? And I'll kind of delve more into my PhD research. So in today's talk, I'm going to first delve in a little bit to where do natural products come from. Then I'll talk more about why nucleotide mimics might be important, and then what total synthesis is in general, and then I'll leave some part at the end to actually go into a little bit of what my completed PhD project has been, which is a total synthesis of the natural product called Ultimocytin. So where do natural products come from? Natural products can kind of come from anywhere. They can come from animal sources. A lot of the ones we study in my lab come from plants, and also microorganisms like bacteria and fungi. And so here I've included some kind of small snippets, and I have a link at the end if you want to actually be able to read these, because I know they're quite small. But so what can natural products do? What are they exactly? And so they're basically a type of molecule, and you've probably seen them represented in these chemical structures before, and they can do a lot of different things. So for example, they can be sweeteners. They can also be toxins. So if you've gotten a rash in the summer from poison ivy or poison oak, or lots of different plants, these are probably due to a natural product that just wasn't tolerated by your skin very well. We've also got toxins in fruits and vegetables that have evolved over time so that fruits and vegetables can defend themselves against insects and the like. And so these are all present in the world around us. And so to summarize, I've got a little summary here, and also very important to my research is that natural products can also be medicines. And so how that factors in can be a lot of different ways. And so I've put in some of the main kind of hallmarks of natural product success stories in medicine on this slide. And so I started on the left with penicillin, which I think almost anybody has heard of because it's such a wonderful story. Penicillin was isolated from mold and kind of allowed us to come from, the dark ages of pre-World War I, where people died of infection. And now in most of the world, it's very rare to die from minor infections because we have antibiotics. And among the first antibiotic was the discovery of penicillin, which is itself a natural product. And a lot of the penicillins today are still isolated from their natural sources. So they're not actually really made in a factory necessarily. There are factories essentially that grow mold, and then we isolate penicillin analogs from these mold. Another very famous in the synthetic community, but maybe lesser known to the public natural product is called Taxol. I believe it's sold under the name Taxol, but also sometimes known as paclitaxel. And this is isolated from the bark of the Pacific yew tree and is also currently, we do not make this from scratch. If you are prescribed Taxol by a doctor, it is isolated from the bark of this tree. So maybe this can make you think about what you consider to be, a chemical versus something very natural. In some ways, even though Taxol is a drug that is prescribed to you, there's something very natural about it because it is really directly isolated from the bark of this yew tree. Sometimes it's modified here and there, but most of the time it's this natural product. And it's been shown to be a very effective compound in treating some cancers. And then the last thing I have on this slide all the way on the right is a compound called cyclosporin A, which is isolated from a type of fungus. And this is routinely taken by anybody who's gotten an organ donation to prevent organ rejection. It's also taken for those suffering from rheumatoid arthritis and actually a lot of other diseases, because this is actually, and I will not be going into this on this talk, but it's a very fascinating molecule which actually can fold. Because it's so large, it can fold and unfold on itself depending on the conditions in which it's in. And it's sort of this beautiful molecule that nature has evolved, and scientists, there's hundreds and thousands of papers in the literature trying to understand how we can make molecules that act like cyclosporin A, because it's just so good at adapting to its environment and doing really amazing stuff to treat disease in the human body. So this kind of forms the basis of why myself and many other synthetic chemists are interested in natural products, because there's this huge potential to use them to treat really important diseases. And instead of having to go through the laborious process of isolating them in nature, which can be very costly for a lot of reasons, we're also interested in sometimes seeing if we can make them more easily in a lab, and down the line, sometimes this can also involve like green technologies and so on. So there's a lot of factors that go into deciding to do this kind of research. And the other guiding principle is sort of what mechanism do we want a natural product to turn into a drug? What mechanism of action do we want a certain natural product to have? And so when I started to decide which natural products I was interested in studying, I looked into an area of nucleotides and nucleotide mimics. So nucleotides are an incredibly broad category of molecules, and they're ubiquitous in living systems. At its most basic, I've shown some of the molecules in this slide. So all the way on the left here, you have something called ATP and AMP, and these stand for adenosine triphosphate and adenosine monophosphate. And these are just necessary for transfer of energy in living cell. They're kind of one of the most basic units of energy in living forms. And then there's this slightly more complex structure that's called cyclic guanosine adenosine monophosphate, or sometimes called CGAMP, which also serves to transfer signals within living cells. And then the other place that you might hear a lot about nucleotides are just that they provide the most basic backbones to our DNA and RNA, among many, many, many other roles. This is a very sort of simplified view of where we find nucleotides. And so how this factors into human disease is that when nucleotides stop transferring signals or begin to transfer potentially incorrect signals, our cells tend to enter disease states. And so, for example, we sometimes get cancer or encounter other diseases. And so this is why scientists are sometimes very interested in being able to mimic nucleotides, because potentially if we can give somebody a synthetic nucleotide, we might be able to turn certain signals back on or turn off incorrect signals and then force the correct signals through so that our body can heal and we can be healthy again. And so this is what motivated my interest in studying the compound all the way on the right, which is called Ultimocytin. In reading the literature, we thought that Ultimocytin might kind of look like AMP, adenosine monophosphate, and that it might act as a nucleotide mimic, so it might imitate nucleotides in some way. And so we thought we would be interested in synthesizing this in the lab, because it would be nice to have a lot of Ultimocytin on hand so that we could study how well it might mimic AMP in biology. Additionally, as a chemist, we thought there were a lot of opportunities for us to also further the field of chemistry itself by potentially discovering new reactions while we were trying to synthesize this big molecule as part of a larger project. So part of the main purpose of this slide is not only for you to sort of see the structure of Ultimocytin on the right, which is actually, it's a natural product isolated from a bacteria, but also to show you how it might mimic AMP. So it's both the 3D structure that we thought might mimic, but also the distribution of charge. And so that's what this kind of rainbow colored cloud around the molecules is. I generated these using a computer program, and the red is more negative charge, and as it gets into purple, it represents more positive charge. And so you can see that there's this kind of like electron landscape around both molecules that has some similarities of where the reds and where the blues are. And so this was the main motivation for me to begin my research on the synthesis of Ultimocytin. So before I, went to my hood in the lab to start trying to make this molecule, and essentially you can think of it as trying to make a recipe to this molecule, I first needed to sit at my desk and talk to my advisor and talk to my colleagues and come up with a good plan. And so first thing I did was I examined the structural features of Ultimocytin. And now for the purposes of this talk, I've mainly depicted Ultimocytin in its 3D structure. I thought this would be a little bit more easier to visualize for our audience. And so the kind of code to read this figure here is that in white you have hydrogen and in gray you have carbon. And then the next 3 types of atoms here, oxygen, nitrogen, and sulfur, are considered polar atoms. And what polar means is that they just tend to have a lot of electron density. And as a chemist, as a synthetic chemist, usually polar atoms are hard to install. And so they were something I thought a lot about because I knew they would all pose various challenges. And so as I went on to make my synthetic plan, I also took note of several structural features. So the first is that Ultimocytin is made up of 2 rings, a 5-membered ring and a 6-membered ring. It also has this side chain here on the side of the molecule. And lastly, as I just mentioned, it has about 7 polar atoms on its main core structure outside of the side chain. So these were all structural features that I was going to have to think very carefully about for how I was going to make the molecule. And so then after making these observations, I went forward to try and make a plan on paper. And so what this process is called is retrosynthesis, which essentially means backwards synthesis. And believe it or not, this simple exercise it actually won a Nobel Prize. And so that's how important this sort of thought process and way of thinking has been to chemistry. And it was not always an obvious way to do chemistry. So it requires sort of thinking backwards. And so how I thought about this molecule backwards was that I thought I could simplify it by removing its side chain, and also by removing this methyl group on the bottom, which is a carbon. And then I thought I could break the 2 rings in half, essentially, because 2 rings was a little bit complicated. And so this would give me what I would call a northern fragment. And then it would leave me with the southern fragment in the center of the slide, which itself is a 6-membered ring. And as a chemist, I realized that this kind of looked like a lot like a chemical that is now shown in this reddish orange color called pyridine. And as a chemist, I also know that there's a related compound called nicotinamide, which is just a type of pyridine with this extra, what's called an amide added onto the side, which mapped on well to the molecule, as you can see. And the good news about this chemical is it's really cheap, it's easy to buy, and it's what we call a feedstock chemical. And it's actually, if you ever kind of look at the ingredients in over-the-counter beauty products or health supplements, it's actually in a lot of stuff. So it's very safe. It's not a particularly toxic compound, at least in small quantities. And so it's very abundant. And so it made a great way for us to start the synthesis, because we don't want to start the synthesis with something super complicated and super expensive and super challenging to get our hands on. So we started our synthesis with nicotinamide. And so with that kind of planning in mind, I was ready to go to my hood in the lab and start making this molecule. So before I go into the forward synthesis, I thought I would give you a quick visual of what my days in the lab look like. So this is me at my hood. As you can see, this photo wasn't particularly staged because it's not particularly clean. But the key components of my work in lab involve wearing a flame retardant lab coat, as some chemicals can ignite. Not pictured in this picture, but I do usually wear lab goggles. So those are always very important in addition to your glasses. I work at a fume hood, which is in some ways kind of like this giant thing that pulls vacuum and makes sure that I do not get exposed to lots of chemicals and solvents. And then it goes through a sort of complex filtration system to make sure that we are not then releasing any chemicals out into the air. I also use a lot a magnetic stir plate and hot plate, so it enables me to stir my reactions instead of, I don't have to do it by hand. This thing stirs my reactions for me and heats them for me. And then I have this kind of complex piece of glassware running along the top of my hood, which is called a manifold. And this delivers vacuum and nitrogen gas to my reactions because some chemical reactions are sensitive to oxygen and water, which are present throughout our air. So sometimes I want to run my chemical reactions actually under nitrogen, which is considered an inert atmosphere because nitrogen is not very reactive. And so with this setup going, I was able to start working on my synthesis. And like many things in life, things don't usually go according to plan. And so the plan is useful because it gives you a framework, but then you have to just start trying things at your bench. And you start realizing as things don't work, you start trying to have to come up with new ideas. As we sometimes say in the lab, necessity is the mother of invention. So, you learn of a new problem and then you have to think really hard to try and fix it. So the plan is on the left here. The plan is I wanted to have this pyridine fragment, which is this ring on the bottom, and then I wanted this northern fragment, and I wanted to try and get them together somehow. But that was not, it turned out to be pretty challenging. That was not working out very well. And so after a lot of experimentation, I realized I actually needed to add an extra atom onto my molecule to sort of serve as an extra crutch or tether, sort of instead of having 2 points of contact, I was maybe going to get 3 points of contact. And so then these two pieces were going to be able to come together more easily. And so that hypothesis turned out to be true. And with this extra oxygen atom on my molecule, I was able to bring the two halves together and form what I call the tricycle because it has 3 rings in it. So 3 cyclic structures. And so with that in place, I then did a next chemical step to install that methyl group I had shown earlier. And so now I was in pretty good shape. A lot of these polar atoms I talked about before were installed. And so things were looking pretty well. But before I could move on to the end of the synthesis, I now needed to figure out a way to actually remove this extra atom, because this oxygen is not in the natural product. And so it had helped me out, but now I needed to sort of edit it out and remove the crutch. And removing atoms in chemistry is not always that easy. We're used to adding atoms on and not usually removing them. And so while this was a frustrating challenge, it was also a kind of cool challenge to have, because it was necessary for me to do this. It kind of stretched my thinking and stretched me to think about what new chemical reactivity I could maybe come up with, so that I could maybe make some new contributions to chemistry. And so after a lot of experimentation, I realized that I could use a reagent based in molybdenum metal. So molybdenum is kind of an unusual metal for this reaction, but I realized that molybdenum metal coupled with some other reagents could effectively allow me to edit out this oxygen and liberate out this nitrogen atom. So now I had this free nitrogen atom, which then enabled me to move forward after a few more manipulations into the last step of the synthesis, which was to put on the side chain that I had shown at the very beginning. And so with another chemical reaction, I was able to lock that side chain on and make the natural product. Overall, this takes about 11 steps. Which is really nice, because this molecule has been made once before in the 1990s, and their synthesis took over 30 steps. And so their synthesis was not very useful for further research. And so we hope that our synthesis is going to be useful for further research. So moving forward, here's my kind of summary slide. First, I'm very happy because this total synthesis, we've been able to report and publish our findings in a peer-reviewed academic journal. So in the world of academia, this is kind of a very nice thing to have. It lets you communicate with other scientists about what you found. And I'm particularly proud of the molybdenum reaction, because I think this will interest maybe some other scientists and see how they might be able to apply that chemistry in their work in the future. Because the synthesis is relatively short at 11 steps, I've been able to now be in the lab a few more weeks and make more of this natural product. So that's what I'm doing actually day to day right now. And then personally for me, this is also great because I get to graduate. And so I will be getting my degree this summer once I finish writing my thesis, writing this work up in a thesis, and move on to postdoctoral studies. So with that, I just have a quick thank you to my lab and to my PI, Professor Thomas Maimoni. I didn't include a link, but if you want to just see more about our lab, you can just kind of Google Professor Thomas Maimoni's name, and our lab website should pop up. I've also been very grateful to receive funding for my work from several different sources, and I'm very appreciative to all of you for your attention and potentially questions. Before I end, I'll also scoot on, just for the sake of the screen share, the image credits and other sources, and then just come back to our last slide. And I also do want to point out, for anybody who's just interested in natural products and synthetic chemistry in general, the Chemistry and Engineering magazine, C&EN, has this wonderful website. And I've linked here with these periodic graphics that give you quick, easy to understand snippets of chemistry in the real world. So thank you.
Great, thank you so much. We're happy to take questions in the chat or Q&A, but I'll start us off. Maybe, I think you said this Ultimocytin is made in fungi, or
this, yeah. Or finish your question and then I'll answer.
Yeah, I guess I was going to ask, like, at large scale, if this does end up being a medicine, what benefits does like chemical synthesis pathways generally have over like natural extraction?
Yeah, that's a very, very good question. Yeah, so this product's made by Streptomyces bacteria, but similar. So, and what you ask is a question that's very important to ask. Synthetic chemists, we tend to like to think synthesis is the best because we enjoy doing it in the lab. But then you have to really think about the reality and what's going to be good for our planet, what's going to be good for everybody, costs and how much you can make and stuff. And so I think the real answer is actually a mixture of both. And so is to be able to maybe have some parts of a synthesis that are made by things like mold or bacteria, and then some parts that we use chemicals. Because one of the downsides of just doing all these extractions is usually they require a lot of water and a lot of organic solvents, because you've got to basically like chew up all the bacteria stuff. The bacteria need medium to grow in. The Taxol I showed at the beginning, the eubark, like they have to like digest all that stuff in these huge vats of water and solvents. And so while sometimes it sounds nice to be like, oh, let's extract stuff from nature, like what could be more natural than that? There's definitely downsides in that way. And so then you've got various branches of research that maybe even have to do with some of the stuff that Evan's done with engineering of plant pathways, because we can try and make the plant even better at making the natural product. We can also try and make like a plant that's able to make part of the natural product in high yields, and then we can do some chemical synthesis on it. So I think that's maybe the best solution. It's part of what allowed both Jennifer Doudna and Frances Arnold to win Nobel Prizes in recent years. It's because I think they're at the forefront of helping us figure these aspects out.
Cool, thank you. Then we've also got this question. Yeah, when you confirmed that you got synthesis of Ultimocytin, how did you feel? What did you do?
I was very excited, but to be honest, it was a relief. I was also, to add a bit of my personal life, I was like 8 months pregnant, so I was really, really excited because I was like, okay, this kid can come and I finished this project. But the challenge of this molecule was actually that the natural product turned out to be very challenging to purify, which is another like thing to do with like trying to isolate things from nature. And to publish a paper, you need like really pure compound. And so I ended up taking some time off from the lab, and my advisor was very generous and very understanding. And then I came back after a few months, and I basically spent all of that time just trying to make a natural product and purifying it, which I have to say to all of you who do research and PhD research, this is probably the lowest time of my PhD research, because it was like, we already made it, like, I want this to be over. Why isn't it pure? Isn't there some kind of machine somewhere that can do this for me? But that, it worked out in the end. We were able to kind of figure out what the problem was and get through, but that was unfortunately a very long process that involved in getting this work out.
And I guess adding on to that, how do you determine you've synthesized it finally? How do you determine that it's as pure as you want it?
Yeah, so our primary analytical method in the lab is something called nuclear magnetic resonance, so NMR. And NMR tends to, how it looks, some of you probably like totally know what this is, and some of you maybe have no idea. How it actually looks is you have like a piece of paper, and there's like a trace on it, basically, with very, very in-depth patterns that we can reproducibly read. It's not just like, oh, there's a peak here and there's a peak here. There's splitting patterns and where the shifts are, and those usually represent where the protons of the molecule are, and they also tell you a lot about where the protons are near. And so every molecule will have a unique one of these, both carbon and proton. And so that's our main thing, is we're able to confirm that those match exactly. And that the good news is they did. The other thing we can sometimes use too, that's a little bit less specific, is you can identify the mass the molecular weight. And that sort of tells you how many nitrogens, carbons, oxygens, and stuff. And so our molecular weight also matches with a lot of significant figures. So that's good. And then those are the two main methods. Sometimes we have, you can have machines that give you these other spectra and things, but my lab tends not to rely on these. We're a little bit old school, so we tend to just rely on the mass and the NMR spectrum.
Cool, great. Well, I think currently there are no other questions in the chat or Q&A. So thank you guys very much. I think our speakers both gave contact details, and of course, Popping the Science Bubble is also online if you have any later questions for these speakers. But yeah, thank you guys very much for being here. These were two very interesting talks.
Such great talks. Thank you so much. Both these talks are available on Facebook and YouTube. Thank you again for attending tonight, and thank you to our wonderful speakers.